Hi everyone and welcome to 9th and 10th channel of BYJU'S. I'm your teacher Ankita and I welcome you all in today's amazing session everyone. Good morning. I hope that all of you are amazing and are doing really very well. Good morning everyone. Please do write in the chat box if you can see me clearly and if you can hear me clearly. Good morning everyone. Good morning. Yes ma'am. Please come.
Good morning everyone and welcome to this class on the 15 important previous year questions on heredity and evolution. I'm also your biology teacher Aishwarya and today both of us together are going to make sure that they ace this chapter. Yes. Because ma'am, most of them they get very worried looking at the chapter, right?
Yeah, especially with the questions that we have over here at this time we feel that, oh, even though we have studied it 1,000 times, we still feel that we need to do more and more practice, and as we all say, practice makes things really very perfect. So that's why we all are here and we'll be discussing these amazing questions with you.
Yes, so good morning everybody and please make sure you have your notebooks and pens ready with you. And of course, we're going to get started and ma'am, I think in about the next 1.5 hours, we are going to wind this class up, right? So we are going to have a total of 15 questions, out of which the first 8 Ankita ma'am will be taking, followed by which the next 7 I'll be taking. So we'll not waste any more time, we'll get started right to it. So Ankita ma'am, all yours.
Yes everyone, so I hope that all of you are sitting with your notebook and with your pencils or the pen, right? So we'll be starting. I'll not be taking much of your time everyone. Yes, we will be answering your doubts. If you have any doubts, you can write over here. Ma'am will be coming back, so you can ask your doubts to ma'am also. Awesome everyone. So now that you have said that we are clearly visible and audible, so we'll quickly start in a bit. If you're new here, if you're watching this video for the very first time, please make sure to hit the subscribe button and please make sure to hit the like button for the video. Now on this channel of BYJU'S and 9th and 10th, especially during your examination, we are making sure that you're exam ready and you're ready to ace your examination. For that, we have different types of classes which are happening on our channel. We have crash courses and along with that, we have one-shots which is already there. We have mock test paper which will be coming really very soon. And we have all sort of different classes that will be helping you to learn better and ace your examination. So I hope that all of you have subscribed to the channel. Please do write in the chat box everyone how many of you have subscribed to the channel and please make sure to hit the like button. Like target wherever I think by the end of this class, we should have more than 300. Yes? Yes, everyone? Hello, everyone. Hello. I'm really sorry if I'm not able to take all of your names. Yes, because there are so many of you and I feel so proud of you that you all are here with us and will be learning together. Yes, I'm feeling good. Thank you so much. I hope that all of you are feeling good. Yes. We don't have Menti quiz today. We will be having Menti quiz later part in the upcoming weeks. You had a Menti quiz last week, so don't worry about it. And you had it yesterday also. Okay, everyone, let's get started. Yes, Hindi mein, so of course, see, in your examination, you will be writing your exam in English. So, we'll make sure to help you in that also. Hindi mein, just in worst case, if you don't understand anything, we might explain in Hindi, but mostly our medium will be in English. Not English, English. Okay, let's get started everyone. So here we have, let's take a look at the very first question. Right? Here we have. Okay, please go and get your notebooks. I'm just giving you 30 more seconds everyone. Right? We'll have 30 more seconds and I want all of you to quickly go. Get your notebook, pen, and yes, and then we'll get started. Yes. So here we have the question number 1, right? And everyone read this question till after that we'll start the explanation part of it. So this question came in the year 2020 and it has 3 parts, right? So let's quickly read the question. Why did Mendel carry out an experiment to study inheritance of 2 traits in a garden pea? What were his findings with respect to inheritance of traits in F1 and F2 generation? And state the ratio obtained in F2 generation in the above mentioned experiments. Easy, right? It's a very easy question. Yes. Very good, yeah. Yes. Nice, very good. I can see the answers. Very good, everyone. Please do write the answer in the chat box. That's very good, Anshul. Thank you so much. Very good. Right. short life, okay. What else? We can easily differentiate between types, okay. Yes, bisexual, okay. But are we looking for that? We're talking about to study the inheritance of two traits in the garden P. Why did he choose, right? Or carry out an experiment to study the inheritance of two traits in garden P. Okay, I can see the answers. Let's quickly discuss this, everyone, right? Now we know that, of course, over here, he actually had a look at the alleles, right? And of course, we know what are alleles. Can you quickly tell me what are alleles? Yes, it was easy for him, for sure. Yes? What are alleles? Very good, very good. So we know that, of course, there are the two different forms, right? The alternative form of a gene. Very good, everyone. Our alternative form of a gene is known as the allele. So, of course, when he was doing an experiment, right, he saw that in this particular experiment, he saw that they're expressing themselves independently, right? Now, of course, you know that which cross we are discussing over here. Yes? Very good. So over here, what are we discussing? We are discussing about the dihybrid cross, right? Now, he took the two important characteristic features. So, of course, the two characteristic features he opted for was a yellow color seed, which is round in shape, and green color seed, which is wrinkled, right? So, why did he carry on an experiment of inheritance of two traits so that he can understand and he can actually see whether they're expressing themselves independently or not, right? That is the last law of the inheritance, which is the law of independence assortment, right? I hope that everyone got this, right? Why he opted for this? See, I can see in the beginning we all were saying that because we were discussing about the P characteristics. But over here in this particular question, what we have to focus is on the inheritance of two traits in the garden P. So this law, when you look at the question, this law is talking about the third law of the law of inheritance, which is the independent assortment. Everyone, are we clear? A quick thumbs up, everyone in the chat. Quickly. See, it's very important for all of us to read the question carefully. Yes? Awesome, everyone. So I hope that all of you are there with me and you got this. Yes, Ashwini, we will discuss about it. Yes. Awesome, everyone. Let's move to the second part of this question. Now, if you look at this particular question, right, we have highlighted the important words. What were his findings with respect to inheritance of traits in F1 and F2 generation? Now, this becomes really very easy. Now, what are we focusing on? It's an easy peasy answer. What about his finding with respect to inheritance of the trait in F1 and F2 generation? Yes. Everyone, what do you think? Very good, very good. Yes, in F1 generation, the dominating genes were shown. Very good, everyone. Hello everyone. Those of you who are joining us right now, welcome to the class. Awesome. Yes. So we know that in the dihybrid cross, we have two traits, right? We are discussing about two traits. Yellow seed, which was round in shape, and green seed, which was wrinkled, right? Now we have to find out what were the outcome or what were the results in the F1 and the F2 generation. So what we'll do, I'm sure you all have discussed, right? Yesterday with Aishwarya ma'am. Yes, very good, very good. Awesome, everyone, right? So if you have watched yesterday's session, so I'm sure you'll be able to help me with this particular question. So we have the two important characteristic features, right? We have the important, basically the genotype of it, you will split it. We have the gametes, we have the dominating one, and then we have the recessive one. Yes, right? We have the capital Y and capital R and small y and small r. These are the gametes. And once we'll see the fusion of these gametes, what we'll see? We will see capital Y and small y and capital R and small r. Basically, the seed shape is round and it is yellow in color. Everyone, are we clear up to this? Satyam, I'm explaining this again. Yes, I'll explain this. Anshul, we will discuss how to frame this particular question in the answer. Yes, Mr. Warning. Yes, Yash. Very good, Vaibhavi. Yes, Ashwini. Yes, Shreya. Very good, musical. Smita. Crystal clear, right? Yes. Okay, Vansh. हाँ, स्टडी किंग, क्योंकि एग्जाम में आपको इंग्लिश में ही लिखना पड़ेगा, है ना? अगर अभी अंग्रेजी में नहीं पढ़ोगे तो बाद में एग्जाम में अंग्रेजी में कैसे लिखोगे? So we have to try now. Don't worry. चलिए, I'm helping you with it. Please pay attention. I'm sure you will get it. Yes. See, your name is in English, so I'm sure you know English. Don't worry, we are here with you. Okay. What were his findings? See, the question is asking that. So once he did the cross, right, with the dihybrid cross, which is focusing on the independent assortment. In this particular question, we have to find out what were the outcome in F1 and F2 generation. Clear, everyone? Everyone, are you clear with the question? What the question is asking? Quick thumbs up. You can write with the pen, and you don't have to cross it, bache. Amma, you don't have to cross it. Yes, everyone, I hope that all of you are clear with the question part. Awesome. Awesome, everyone. Yes, we will make the Punnett square. Don't worry, we'll make it. Okay. Now, see over here what we have. Yes, it's okay. We are going really very slow. Don't worry, we are going really very slow. So you will not miss anything. Okay. Yash will discuss about the segregation in a bit. So over here, if you see, let's start from the very beginning, everyone. So he took a plant, right? He took a plant with the two important characteristic features for our understanding. We have a yellow and a round seed. Everyone, are we clear with this? Right? He took one plant which has round seed which are yellow in color, and he took one more plant which had the green color seed which was wrinkled. Yes? Milega PDF, you will get the PDF in the Telegram. Yes. Now, what we do, we will see the genotype of it. Over here, this is a dominating characteristic. So, we will write in this particular way. Capital Y represents a yellow color and capital R represents the round shape of the seed. So, these are the dominating characteristics. That means that they will express themselves, right? Then, we have small y and small r. That means that these are recessive one and they will not be able to express themselves in the presence of the dominating one. Clear? Yes, very good. This is dihybrid cross. Absolutely correct. Okay? Dhanya, we will. Blueprint as such we don't have, but yeah, we have done various shots on that. So you can go and watch that. All the best ones for your examination. Everyone, are we clear, right? Yes. Send us a chart and let it denote it to them. I'll share, Yash. Okay. Very good, very good. So what we have over here, so we are clear with that. We have the phenotype and we have the genotype. And what we'll see the gametes. Very good. Right? Yes, we have the gametes now. Now in gametes, what we have the capital and the recessive one. See over here, I'm sure all of you now know that the capital letters, right, will be representing the dominating trait and the recessive one will be represented with the small alphabets, right? So what we have the gametes also. Everyone, are we clear? We can Aditi, but in this particular question, it's very easy for us to do it with this particular cross only. Awesome, everyone. So everyone, after the cross in F1 generation, what we have? In F1 generation, we have capital Y, capital R and small r. There are two gametes, so they will be coming together. And what we have in the F1 generation. So in F1 generation, everyone, what we have a yellow seed, right? Yellow color seed, which is rounded shape. Clear? Siddhi, I'm explaining this question again. Please pay attention. 404, this is dihybrid cross. We will have social study sessions also in the future. Okay. Aman will have it today in the evening. Okay. Everyone, are we clear up to here? Yes. Very good, everyone. So we are clear that in F1 generation, all the seeds will be yellow in color and round in shape. Easy peasy? Yes. So that is the first part of the question. Now let's take a look at the second part. What will be the result in the F2 generation, everyone? Here we have Genotype in F1 generation was this only. Yes. Now everyone, let's take a look at the F2 generation. So in F1 generation we got this, right? We'll do the selfing of it. Yes, we'll do the selfing. And after the selfing, what we'll have? We'll have this. I'm sure as you all are mentioning. Yes, 9:3:3:1. So what will happen over here? In F1 generation, we will do the selfing capital Y, small y, capital R and small r cross with the same capital R, small y and capital Y, small y, capital R and small r. After that, what we'll have? We'll have this as a result. Yes, I'm sure all of you have seen this. And once the cross is done, we will be able to see the ratio. 9 are yellow in color and round in shape. 3 are yellow in color and wrinkled in shape. 3 are again green in color, right? And round in shape. And 1 which is green and wrinkled. Everyone, this is easy, right? Yes, this is easy. I am good. Thank you for asking. I hope that you're also good. This question comes for 5 marks. Thank you, Vaishnavi. Easy peasy, right, everyone? See, so I'm telling you, everyone, you don't have to worry about these questions, right? These are very easy questions. Aapki textbook mein you have the dihybrid cross. Solve that cross 1 or 2 times. It's really very easy. If you want, we can start it again. See from here. We started with this, right? We started with this. We have these 2 important traits. We will be representing them with a capital Y, right? And capital R, small y and small r, right? We will have the gametes, that is capital R. And we will have this. Once we fuse them together, we will have this. This is our F1 generation. Easy peasy, right? We'll do the selfing of this and we will get the result. Yes? Good, everyone. Ma'am, is self-fertilization and self-pollination is same. So self-fertilization and self-pollination, we will discuss it out. But just look at the word self-fertilization and self-pollination. You will have the answer with you. We'll discuss in a bit. Give me some time. Segregation, yes, over here means that. Over here we can see, right, there's a segregation. The gametes got separated out, right? Yes, the law of segregation says that during the gamete formation, each gamete will get separated. Okay? Yes, I hope that this is clear. Yes, Ajit, F2 will have different characteristic features. Absolutely correct. PG, this time, yeah, it is a little bit difficult, but don't worry, we'll be solving it today. Today, right? So we'll be solving it. Everyone, quick thumbs up. Yes, counting on the phenotype ratio is a waste of time. No, it's very easy. 9:3:3:1. Genotype, they haven't asked us, right? In our class 10th, they haven't asked us about the genotype. So that if you're doing that, that could be too much time taking, but the phenotypic ratio is really very easy. 9:3:3:1. Very good, everyone. So I hope that this is clear to each one of you. Yes? See, we have explained this question now 5 times. Yes. Very good, Siddhi. Very good. You can draw with the pencil only, Aman. See, if you draw it with a pen, and if by mistake, if you write something wrong, it will be difficult for you to rub also, right? Then you will be cutting it out. It's good if you draw it. If you draw this with the pen, you can easily erase, with the pencil, you can easily erase it also. Okay? Everyone, are we clear? Quick thumbs up. Come on, come on. How's the Josh? Yes, Anshul, in a minute we are starting. Sachin, Bachche, this session will be hopefully for 1.5 hours. Hi, Shri Hari. Sai Hari. Very good, very good. Good to see that all of you are giving me thumbs up and Josh is really very high. Let's take a look at the C part of this question, everyone. Here. Genetic makeup we will be discussing about basically the, I'll tell you what is the genotype also. Yes, in a minute. Yes, Afreen, welcome. Yes, Vaishnavi. Okay. C part, everyone, state the ratio obtained in F2 generation in the above mentioned experiment. Easy peasy. What was the ratio obtained, everyone, in the above cross? 9:3:3:1. Super awesome. See over here what we have is the phenotypic ratio. Okay, very good everyone, very good. Now, Yash, you were asking about the genetic makeup, right? So for example, if I have to see, this is a genetic makeup, what is actually there, right? I know that, okay, these three are wrinkled in shape and yellow in color, right? But they have different combinations. Okay, see if you look over here, see this capital Y, it has two capital Y, it has only one capital Y, and it has again one capital Y. So what is the actual genetic makeup? What types of traits they have, right? That we have as a genetic makeup. Yes, Aditi, we don't have the genotypic ratio for this particular experiment. Okay? That's nice. Everyone, I hope that you got it. Satyam, we don't have Menti today. Soham, let's not spam. We don't have Menti today in today's class. We'll have it the next week. Come on, don't worry. We have amazing quizzes coming on your way. Okay, everyone. So, see, this is how you can write the answer in the examination also. See, the first question was talking about why did he carry out this experiment? So, you can mention that he carried out this experiment so that he can understand the interaction that is happening between the two different traits, right? And we can mention about, over here, we can mention that in dihybrid cross, it observed that the two pairs of the characteristics were considered and they will be expressing themselves independently. Are we clear? Yes, they're expressing themselves independently. Megha, so the ratio will be 9:3:3:1. Easy peasy, bache. Okay, everyone? PDF, you will get it after this class. The second part of this question, everyone. What was his finding in F1 and F2 generation? So you can easily write it. Right? In F1 generation, all plants were round and yellow seed. But in F2 generation, we have this. We have round yellow, round green, wrinkled yellow and wrinkled green also. Awesome. And the last part of this question, we'll just write the ratio. 9:3:3:1. A 5 marks question, everyone. Super easy question. And I'm telling you, see, can I tell you, right? In this particular chapter, there are two important cross. And there are only two cross. Monohybrid and dihybrid. So we have to focus on them, right? Yes. H ambition, the genotypic ratio of the dihybrid cross is really very long. It's very long and we don't have it in our syllabus also. So we'll not be focusing there. Yes. The phenotypic ratio we don't have of F1 generation, we just have one. Right? All were round and they were yellow. So the physical characteristics were round and yellow. Thank you, Manoj, for helping your friends out over here. No, we don't have. We do have, yes, very good. Anshul, we do have, but you will be learning about those crosses in your higher classes. Okay. Everyone, are you ready for question number 2? Come on, come on. Are you ready for question number 2? Yes. And how many of you have hit the like button? Yes. Very good, very good. Awesome, everyone. Very good. Super good to see that all of you are clear with these, right? Okay. Now, let's take a look at the question number 2. Here we go. Now, again, these questions are really important. It's a 5 marks question. And in this particular question, we'll be looking at the monohybrid cross. So, we have just had a look at the dihybrid cross. Now, we'll be taking a look at the monohybrid cross. Okay? Let's, everyone read the question. Vishnu, I'm just increasing the height of the table. Please kindly fix it. It's uncomfortable over here. Yes. Yes, everyone. That's nice. Very good. Okay. So, we have this question, right? We have this question. So, we have a green stem rose plant denoted by the capital G and capital G. That means it's a very, it's the, basically it's a dominating characteristics, right? I'll move it. Yes. Okay. And then, of course, what we have? Yes. So, we have this capital G, and then we have a brown stem rose also denoted by small g and small g. So, what we have in the question? Let's quickly write over here. Right? It's a green stem rose and small g, brown. Can you tell me which one is dominating and can you tell me which one is recessive? Are you giving me answer? You're super fast. But for all of you, can you tell me which one of these is dominating and which one of these is recessive? It could be Afreen. Very good. So this of course we know it's dominating, and of course this we know is a recessive. Okay? Yes. Now we have these two set, right? We have part A and part B. In part A we have 3 questions, so we will be looking them at individually. So let's start with it. So the part A question states that we have to list the observation regarding the color of the stem in their progeny in F1 generation. What do you think? So what we have the capital G, which is the green stem rose plant. And then we have small g and small g, which are brown stem plant, right? Brown stem rose plant. If the cross is happening, we know that what will be the case? We will have the gametes as capital G and the small g. And if you fuse them, what we'll have? We have capital G and small g. So in the F1 generation, all will be very good. So all will be green, right? All will be green stemmed rose. All plants will have the green colored stem. Easy peasy. See, this is a cross, right? And I think many of you were asking about the Punnett square also. So see, this is how you can draw this. So we have capital G, small g, and I hope that all of you got this. See, this is how you can draw the Punnett square in the examination if the question demands. See over here, heterozygous, super awesome, very good. See, I'm very proud when you use these words like heterozygous green. Awesome, everyone. You don't know me and Ashwara ma'am will be super proud when we see these comments. Yes. Awesome. So we are clear with this part, right? That is green stem rose. So first part is clear to each one of you. Awesome. Now let's move to the B part of this question. The B part of this question is that percentage of brown stem plant in F2 generation if plants is self-pollinated. So till now what we have seen in the F1 generation, we got capital G and small g. Yes? If we do the selfing of this, what we will have? If I make the Punnett square, we have capital G, capital G and small g. So capital G. Yes, very good. I can see the answers already. Right? So what we have, the ratio that we will get is to 1 is to 2:1. This is a phenotypic ratio. Right? Sorry, this is a genotypic ratio. Sorry. Okay? When we talk about the phenotypic ratio, is 3:1. So, now what we can easily conclude from this, the question is asking us the percentage of the brown stem tree. So, of course, the brown one will be the one which is recessive, homozygous, recessive. Awesome, everyone. That will be the small g. So, the percentage, if we say out of 100, will be 25 because only 4 are there. We have 4, right? 1, 2, 3, 4. So, out of these 4, only 1 is homozygous recessive and that is the small g and small g. So, are we clear, everyone, the percentage? Homozygous, recessive, that will be the brown stem rose plant. Very good, Vaishnavi. We don't have evolution in our syllabus. Mannat, we don't have. See, smiley for you, everyone. A Kaju Katli also. Chaliye. Let's move to the C part of this question. Right, the third part. Ratio of capital G and capital G and small g in F2 progeny. Let me quickly make the Punnett square again so that all of us are clear with this. Can you quickly help me with the answer, everyone? See, I'm making the Punnett square. We just did this question. Okay, I can see the answers. Yes, The ratio will be 1:2. Absolutely correct. Why the ratio is 1:2? We know that capital G and capital G we just have one time. See over here, we just have one time. And the capital G and small g, we have it two times. We have it over here and we have it over here. So that will be 1:2. Easy peasy. You don't have to, Aditi, you don't have to write the names. When you're making a Punnett square, you don't have to write, but if you're writing the answer, if you have time, you can write. Alam, what you didn't understand, can you explain me? If you can write it, I'll be able to explain you again. So in this particular question, they're asking, capital G, how many times we have? Small g and capital G, how many times we have? Like how many plants have this? Okay? Yes. Awesome. So we have this, right? I hope that it's clear. So this is how you can make the Punnett square. It's super easy. Yes. So that's what. Yes. Okay, you are saying that I am wrong because there are 3 capital G. Okay, let's just figure it out. If you read the question again, they're asking that ratio of capital G and capital G. How many times we can see capital G in the square? See, we have 4, right? We have 4. Everyone, those of you who have some doubts, let's look at over here. How many times in this square you can see capital G and capital G? 1 time, so 1. And how many times you can see a capital G and a small g? How many times you can see a capital G and a small g? 1 or 2 times? 2 times. So, we all are correct. The ratio will be 1:2, right? 1 time we have capital G and capital G. 2 times we have capital G and small g. So, we all are correct. Awesome. That's amazing, everyone. I hope that you all got this. Yes. Awesome, everyone. Yes. Very good. Simple dimple. Nice. Okay. Now, let's see how you can represent this particular question in your answer, right? In your answer sheet. But before that, we have one more part, everyone. So, again, if you look at this particular part, the question is asking, based on the finding of this cross, what is the conclusion that we can draw? Easy peasy. So, we have discussed about this whole monohybrid cross. So, what will be the conclusion that we will be giving? Homozygous tall, yes, Anshul. Afreen, we will not be discussing test cross and back cross here. It's not in your syllabus. But if you want to discuss that, you can go and watch our previous session. We have discussed in those classes. Right? So everyone quickly tell me, what is the conclusion that we get after this cross? We got a conclusion, very good Ashwini, that dominating traits will be expressing itself and recessive traits will only express themselves in the absence of the dominating trait. So we can say that it's all about the monohybrid cross, right? And of course, it's the first law of inheritance, law of dominance. Vaishnavi, just check your screen. Our screen is clear. It's not blur. Okay? So in the heterozygous condition, the allele which expresses itself physically is called as dominant, and other which remain unexpressed is called as recessive. The law of dominance. Hidden character express, we can say that, but when we're talking about, when we're talking about definition, we don't want to write the hidden characteristics, right? We don't want to state in that way. This is how we'll be writing. Yes, Alam, dominating characters are the ones that will be expressing themselves. Okay? Very good. Like black color hair, right? They'll always express them as compared to the brown hair color. That's an example that I'm giving you. Okay. So everyone, this is how you can write this whole question in your examination. See how beautifully it's written over here? You can definitely write in a similar way. You'll be focusing on the parts. Acha. Okay, everyone raise your hand. Kisko issues hote hain when you write these A, B, C, D parts, right? There are times we sometimes miss the parts. Like when we're starting writing with the A part 1, maybe we'll skip the A part 2 and we'll go right on the A part 3. please make sure in the examination you're very, very vigilant about it. Very, very careful about it, because if you do that, you will be losing your marks. Okay? Oh, this is a font. Yes, over here we have, so color of a stem is in F1 generation is a green color stem, that is capital G and small g. You can write in that way. Then we have in F1 generation the progeny, right? You can express, you can make the Punnett square. See, everywhere, it's really important for us to understand that if we have a 5 marks question, please make sure to make a Punnett square. Yes? Study network, this is how you'll be writing the answer for monohybrid cross if the question comes to 5 marks. Yes? So everyone, please make sure to make the Punnett square, especially in monohybrid cross. Underline karna is equally important, right? Very good, very good. Okay. So you can mention what will the percentage of it, and the last part, right? You can clearly see over here, we have highlighted the important words. So see, we are making your job and your writing really very easy. When you download these notes, you will understand that we have highlighted the important keywords also. So you know that what you have to highlight in the examination. Okay? Please make sure to use a correct word, homozygous condition, heterozygous condition, dominant, recessive, right? Yes. So I hope that everyone, are we clear with this? Quickly, a quick thumbs up everyone. Aditi, for the monohybrid cross, it's really very easy. We just have the monohybrid cross, F1 generation, F2 generation. You can easily draw it. Yes. Awesome. We are clear with it, right? So, half an hour up everyone, and we are done with 2 questions. And these are the 2 main questions that we started with. I know it's nahi hai aapki text mein. If you want to write, you can. But if you write, it's an extra advantage that you have. Yes. Okay. So everyone, so we are done with the 2 major questions. Now I want your help to solve these questions really very fast, because we have covered the major part already. Avika, we will try our best. See, I'm speaking really very slowly in English also, right? So, please stay here. Try to understand. We will help you in understanding. I know, Mannat, dihybrid cross looks a bit difficult, but see over here, we have made it super easy for you. Yes, if you practice it, will be easy for you. Yes, scientific name of pea plant. See, your friends are mentioning. It's Pisum. See, I always remember this name because Pisum. Right in South, we have the amazing kheer. जिसे हम हिंदी में खीर बोलते हैं, over here we call it as a Pisum. It's really yummy. So try to relate it with it, please. But spelling should not be wrong. That's really, really very crucial. So even if you don't know how to pronounce it, please make sure you know the correct spelling of it. Right? Know the correct spelling of it, and then you can start working on the pronunciation of it. The pronunciation is also really very easy, but in the examination, it will be getting focused on the what you write. Okay? So here we have, as you already are mentioning, so I'll not be spending much time over here because you already gave me the answer. So the plant which Mendel used to perform his experiments is a pea plant, and the scientific name of the pea plant is Pisum sativum. Okay, please make sure you write the spelling correctly. And even though you will write in the English pea plant, please write the scientific name also. It's really, really important, because pea plant, there are different varieties altogether. On which particular thing he worked, this is a one. So please make sure you write this. Okay, scientific name will give you 1 marks. And if you write pea plant, you'll be losing your half marks, everyone. Yes? Very good. Avika, I will try. Yes? Yes, thank you so much, Vaishnavi. Yes, very good, very good. Chaliye. Everyone, let's take a look at the next question. Okay, here we go. Next question, everyone. In this particular question, what type of progeny was obtained by Mendel in F1 and F2 generation when he crossed a tall and a short plant? Write the ratio he obtained in F2 generation. Can you tell me which cross we are talking about? Yes, which cross we are talking about? Very good. We're talking about the monohybrid cross. Absolutely correct. So, if we have to draw, we have capital T and capital T, small t and small t. Here we have the Punnett square, everyone. Get used to making these Punnett square, and you don't need a scale. Use your hands, right? If you have time, then of course definitely use the scales. But you can quickly make this, right? And over here, capital T, small t, right? Capital T, small t, capital T, small t, and capital T, small t. In F1 generation, all will be tall. Right? Capital T and small t. Yes, very good, very good. Now, what will be the case in F2 generation? In F2 generation, if we do the selfing, we'll have one capital T, then we have one heterozygous tall and one dwarf. So 1:2:1. Frozen creation. How do germ cells make a single set of genes? Interesting question. We'll answer this question. Give me some time. Abhi nahi. We'll let this finish this particular question. Very good. See, I'm very proud of you, everyone. See, if you are clear with one thing, one question, you will be able to solve everything. Okay? So this is how you can write. You can mention about the tall plant, right? You can mention about the genotype. You can mention about the gametes. Then you have the Punnett square, and this is how you will have your answer. Easy peasy, right? So in the F2 generation, what we have this. 1:2:1. 1 is pure tall, 2 are heterozygous tall, 1 is pure or homozygous dwarf. Phenotypic ratio will be 3 are tall and 1 is dwarf. Are we clear? Clear, everyone? Savnath, you have to write, if the question asks you for the genotype and phenotype, then you have to write both. Okay? If the question haven't asked anything for you to write, it's good if you write both. Yes. For the monohybrid cross, both the ratios are important. Genotypic ratio and phenotypic ratios both are important. Yes, these are sufficient for your board examination. This will be helping you to understand what type of question that can come in your exam. Okay? Very good. Yes. So yeah, let me tell you the difference between the genotypic and the phenotypic ratio. Acha. Okay, you tell me this, everyone. Capital T and capital T. Okay, by looking at this, what you can say? Okay, by looking at this particular thing, what will be the conclusion that we will get? Yes. All are tall. Okay, we can say both the plants are tall. Okay. Vaishnavi, it will be 25, bachche. 75 and 25. Yes. Very good. Very good. Frozen creation. Both are tall, but both are different from each other. See, we can say both are tall. And the ratio will be 2, right? Because there are 2 plants which are tall. Both are tall. But if we have to look at the allele, one is homozygous tall. Right? It is homozygous tall and this is heterozygous tall. So this, when we go into the homozygous and heterozygous, this is genotypic ratio, right? This actually help us to understand the genotypic ratio. Are we clear? Yes? जिनको समझ में नहीं आया था, are you clear? Genotypic ratio and phenotypic ratio क्या है? Genotypic ratio वो ratio है जो आपको बताएगा कि उनका actual genetic makeup क्या है. Capital T या फिर capital T and small t. Phenotypic ratio actually help us to understand the physical features that we quickly see. Awesome. So we're done with this, everyone, and this is how you can write your answer in the examination. Danya, 9:3:3:1 is a ratio, phenotypic ratio of dihybrid cross. So this is how, everyone, you can write the answer in the examination. The plant's name, right? The tall pea plant, right? Tall plant, capital T and capital T we will mention. Dominating and recessive trait, and here we'll draw the cross. How to identify gametes and parents? Vinayak, so of course when we start, see, these are the parents. Right? So if we have these parents, right, the gamete, that will be giving us T. And from here, small t. So these two are gametes. Ram, homozygous means same. Can you look over here? Ram, this is homozygous tall and this is homozygous short. If we have capital T and small t together, this is heterozygous. Homo means same. So they have same allele. Hetero means different. So they have different allele. Everyone, are we clear? Come on everyone, this is easy peasy. Quick thumbs up. Now how many likes we have? And how many of them are there in the class? Oh, everyone, come on. Let's make sure to have the equal amount of likes. Everyone quickly hit the like button. All of you who are here in class, please make sure to hit the like button. See, we are going really very slow. Right? My name is Ankita. We will discuss about the law of segregation also. Yes, we are going really very slow so that all of you are understanding. Yes, please make sure to hit the like button, everyone. Yes. Okay. Nice. See, before we finish this, we should have 150 likes. Before I finish this part. Then, next 150 likes is from Aishwarya ma'am. Yes. Okay. Let's move ahead, everyone. But before that, I think you have a doubt on the law of segregation. Now, the law of segregation, you have all the doubts. See, we will show you. We have the proper definitions, right? But if you have missed it, you can go back. Yesterday's session which Aishwarya ma'am took. Ma'am have clearly defined basically the definitions of these law. Yes. We'll show you again, don't worry. Yeah. Note the gametes of capital T and small t will be capital T and small t. Okay. Now let's quickly discuss about the law of segregation. What is law of segregation? What is the meaning of segregation, everyone? Quickly. What is the law of segregation? What is the meaning of segregation? Jaldi batao. Segregation ka matlab kya hai? We all know the meaning of segregation, right? I'm sure your parents would be asking you to separate the tomatoes and the potatoes. Right? Separation. Separate karna. So according to the law of segregation, during the gametes formation, right, during the gamete formation, we will see that gametes will segregate. Okay? Are we clear, everyone? Are we clear? This is the law of segregation. That means that during the gametes formation, we will see the gametes getting separated. Okay, that is law of segregation. And what is law of independent assortment? Law of independent assortment says that even though, right, they are independent, right, after the segregation, they are independent and they will be expressing themselves independently. Yes. Very good, very good. So this is law of segregation where we can see the splitting of the gametes, right? And the law of segregation says that, of course, even though these are getting separated, right, even though they're together in the beginning, they'll express themselves independently. See, nobody is ignoring you. Yes, if you have a valid question, we will definitely answer your question. Okay. Everyone, let's take a look at the next question, question number 4. And this is super easy. Does not have to do anything with any of these tables. But can you quickly help me with this question, everyone? How did Mendel's experiment show that different traits are inherited independently? Yes? Got your point? Goswami, please write this in the comment section. We will definitely come up with that. Don't worry. We'll have it for English also. Very good, very good. Yes? So, very good. I can see the answers, everyone. So, in this particular experiment, when we saw the word independently, right, it's easy for us to understand that this question talks about the dihybrid cross. Yes. I will share, Shiksha, I will share the definitions, right? We have the definitions of all the law. Don't worry, we'll share. You can take a screenshot of it also. Don't worry, we'll share that with you. Yes, full form of PYQs, previous year question paper. Questions basically. Very good everyone. So see, in this particular question, what you have to do? You have to write the dihybrid cross things, right? Yes, very good. So over here, everyone, we have, right? So this is how the gametes are formed. It's basically nothing but the repetition of the dihybrid cross. So here we have, this is how you will write. You will make the cross over here. See everyone, it's a 5 marks question. So you have to make the cross for it. Okay? And if you make this, you are sorted. Then you can write about what you have. And you can mention about the law of independent assortment also. And you will have 5 marks in your pocket. Okay, everyone, are we clear? Are we clear with this? Aisha, we will be doing the mock test paper so that you have a good practice. Yes, study network. You can just make the cross, right, and you will have the answer. And you can write what was the conclusion from that particular thing, and your answer is sorted. Very good. Awesome, everyone. Yes, awesome. Very good, very good. So everyone, are we clear? We must write this and draw the table, right? See, again, see, there are 2 things. See, making a table is really very easy, right? So you can make the table, write few points about that are the highlights, right? There's an independent assortment. We can clearly see, you can mention about the independent assortment. You can write about the result. They have expressed themselves independently and you are good to go. Ram, I didn't get your question. I hope that everything is clear. Yes. Okay. Let's take a look at the question 5, everyone. Here we have question number 5. Easy peasy question, everyone. So over here, what we have? Oh, this is super easy, everyone. Ram, you're saying, ma'am, NCRT exemplar, NCRT. I didn't get you. You're asking about whether this question was from NCRT exemplar or we have the questions from the NCRT exemplar. We had many sessions where we have discussed these questions. So don't worry, you can go back and see those questions. We don't have mentee today. Yesterday we had a mentee class. It is very necessary to solve the practice session. That's why you have to come to today's class in the evening. ऐश्वर्या मैम भी आएंगे जल्दी से. अगर हम ये सारे क्वेश्चंस खत्म करेंगे तो मैम जल्दी से आएंगे. Yes, you want to do the exemplar? Don't worry, we will have it. Yes, stylish gamer, love you too. Love the heredity. Yes. Oh, I see. As a teacher, we all love you. And when you write, ma'am, that ma'am, we love biology. We feel super happy. We feel that wow, it's a very good subject, right? So yes. Good, everyone. Chali, everyone. Easy peasy question, everyone. Ma'am, can we write on the board examination during any hour highlights about the question? I didn't get your question. Can we write on a board question paper during? No. Please do not write anything on the question paper. If this is what you're asking, Manat, please do not write anything on the question paper. Very good, Aditi. Musical shorts, P. P for previous year, Y for, year, Q for questions. Previous year questions. Yes. Avika, let's focus in class, and Dinesh, you also. Your board examinations are here and they're really very important for all of us. Okay. We have to list 2 differences, right, between the dominating trait and the recessive trait. That's the first part of the question. Let's take a look at the first part. Here we go. So the first part of the question, everyone, we have to differentiate, right? And it's an easy question for 2 marks. The differentiation can come easily for 2 marks. And this is how we can write the answer. Dominating traits are controlled by the dominating allele. And the recessive traits are controlled by the recessive allele. And they'll express themselves both in the homozygous and the heterozygous condition. Whereas the recessive traits will be able to express only during the homozygous conditions. Okay? In the heterozygous condition, they'll not be able to express themselves. Are we clear? Yes, for frozen creation, yes. Okay. Okay. Anshul, but in the board examination, please make sure you're not writing anything on the question paper. Right? The examiner might question you. Okay. Yes. So see everyone, this is how you can write, and let's take a look at the next part of this question. What percentage portion of a plant in the F2 generation progeny were round in the Mendel cross between the round and wrinkled? Easy peasy. Right? Yes, see musical shot, I understand it in examination. What happens because of the pressure, we could say, we sometimes what happens, we become really very nervous and we don't read the question carefully. So let's just make sure we are reading the question carefully, right? Take a deep breath. If you feel that you don't know the question, take a deep breath, read the question 2 or 3 times. I'm sure you will have the answer with you. So let's take a look at this question, everyone. So the question is, what we have to find out, what is the percentage ratio of a plant in the F2 generation which were round, okay? In F2 generation, mein kitne round the? Easy peasy question, right? How many of them were round? So it's an easy question. So let's see over here, we have this. And yeah, we have 75, right? Majority of them were round. 25 were the 25% were wrinkled. Clear? It's a monohybrid cross. Yes. Very good, everyone. Okay. Okay, here we go, everyone. So the dominating trait is this, and of course, the total percentage will be the 75. Yes, are we clear? It's a very easy question. I hope that you got this answer. Okay. Let's see, everyone, question number 6, and this is super interesting. Okay, I want all of yours help. Come on, come on. See, this is a very tricky question. See, look at this question. It's very tricky. Given below is an experiment carried out by the Mendel to study the inheritance of 2 traits in garden pea. See over here. Look at the question, everyone. Everyone, take a look at the question. I'll be back in 15 seconds. Just drinking water. Yes or not? I can see you're writing, it's very easy. I'm trying to scare you and you're saying, ma'am, it's easy. Very good, everyone. So, what we have over here, we have the round seed, which is green in color. And we have wrinkled seed, which is yellow in color. See, is tarike se, you can have the question in your examination where they have flipped it, right? So, let's see what we have to write over here. Okay, we have to fill these blanks, and this is the question that we have. So, let's take a look at the first part of this question. So, we have to fill in the box with the appropriate answer. We have to write the gametes. What will be over here? Capital R and capital Y. See over here, capital R and small y. And over here, small r and capital Y. Are we clear? Right? These are the gametes, right? We can easily get the gametes from the parents. One will take easy. Yes. Then, of course, when we do the cross of it, we have a round and the yellow seed because these are the dominating characteristic features, right? And once we do the selfing of it, what we will have? We'll have this, and the ratio will be 9:3:3:1. Easy peasy. Yes. Super awesome, everyone. See, this is what we have. Capital R, capital Y and small y. Easy, ma'am. Is it? Ram, see, bache, yahan pe we have this particular chart, right? So what we have, they have given this, these are the parents, right? We have to find the gametes. So what will take, we have 4. See, in very easy way, if I have to explain you, there are 2 characteristic features. And there are 4 alphabets that are written over here. We have capital R and small y. Though there are 2 capital R and 2 small y. So we know that 2 are same. So we'll just take capital R and small y. And from the other one, we'll take small r and capital Y. Once we'll fuse it, we'll have capital R, small r, capital Y and small y. Once we have the fusion of these, then we'll have this particular ratio. Okay? Okay, let's move ahead, everyone, to this particular part now. Yes. Here we have, why did Mendel carry out an experiment with two traits? Now, this is a very interesting question. We have it in the first question also. Okay, why he thought that he should perform an experiment with two traits? What was the reason behind he opting for two traits? We already have answered this question, right? We know that when he took the two traits, he actually understood about the independent assortment, right? We saw that alleles of two different characteristics or traits separately, they basically segregated independently, right? It's a third law of the law of inheritance, that is nothing but the law of independent assortment. Yes. Don't worry, everyone, I will be sharing the question, this whole PDF with you on the Telegram. Don't worry. Yes, very good, everyone. So that's a B part. We are done with it. Here we go to the C part of this question, everyone. What were his findings with respect to inheritance of the trait in F1 and F2 generation? What do you think? Easy, right? Can you quickly help me with the answer, everyone? What was his finding with respect to inheritance for the first and the second generation? First generation we know that all were round and yellow. Right over here, we can see they were round and yellow. And in F2 generation, of course, the ratio was 9:3:3:1. Very good, everyone. See, it's all interconnected. See, my children, you all are my dear bachas, and I'm telling you, and I think Ashira ma'am will tell you this. Both of these crosses are really important, dihybrid cross and monohybrid cross. These are the only two cross you have, and it will be coming in a different way in your examination. So the more you practice the questions, the more clarity you have, and you know that what you have to write in the examination. So usually it will be easy, don't worry, Mannat. Yes. Musical shorts will take that question. 1 minute, give me some time. Okay. Harshita, in that case, you can take the screenshot. Very good. So here we go, everyone. This is how you can write the answer. It's a very easy question. Okay. Question number 7, everyone. Vaishnavi, I'm fine. Don't worry, I'm fine. Yes. Here we go. Avika, you wanted to explain this question again. So the question is very easy. So you will, in the examination, they'll give you a structure like this, right? They'll give you this table. Right, and based upon this table, they'll ask you to find the answers. So if you look at this table, by looking at this only, you can find out that it's a dihybrid cross. So your answer becomes really easy. Okay? Yes. Okay, let's take a look at the question number 7, everyone. This is really easy. Yes, this is super easy, right? Can you tell me what are we discussing in this particular question? Again, a very important concept that we have in heredity chapter. And this, of course, comes from the how do organism reproduce also. The sex determination, right? It's a very important concept. And 3 marks question, it's kind of sure shot. So, yes, so we have to identify the male and female in the given figure. And then, of course, we have to identify 3, 4, 5, 6, 7, 8. It's very easy, right? So, what we have over here, X and Y, XY will be the male and XX is the female, right? Yes, male have X and Y chromosomes and female have X and X chromosome. If the cross will be there, we'll have X, there will be 2 double X, right? And 2 X and Y. Yes, very good, everyone, very good. So it's really very easy. We know that in the male, they have 2 different types of sex chromosome, right? They have X and the Y, so sperms will be having one. It could be having the X chromosome. The other can have the Y chromosome. And if the X chromosome, right, which is the sperm which is carrying the X chromosome, fuses with the female X chromosome, the gender of the child will be female. But if a sperm carrying a Y chromosome fuses with the egg which has X chromosome, we will see the gender of the child will be boy. So it's a 50-50% probability, and males are responsible for the determination of the gender, right? Not the female. So everyone, this is how you can frame your answer. Yes. Frozen, what is autonomous? So of course there are two types of chromosomes that we have, right? We have sex chromosome and then we have autonomous chromosome. Basically, throughout our body, in all our cells, right, we have, for example, on skin, we have autonomous chromosomes. Okay? That's how we say, because these are the ones that will be helping. There are only one sex chromosome, rest all. From 1 to 22, we call them as the autosomes, right? They will be helping and giving the different characteristic features like skin, different altogether, whatever we have in our body. Then, of course, the sex chromosome will be helping us to have our gender identified. Yes, very good. We don't have evolution. Yes. Here, I'll show you the question again. So, the question is this. So, we have to identify the given blocks that we have. Okay? Very good, everyone. So, are we clear? Harshita, for that, you have to download the Telegram, bache, for the PDF. Clear, everyone? Prakha, thank you so much for helping us with this detail. But let's focus on biology. Okay. That Y gametes is going to extend. Mritin, I'm not sure. Let's hope. Right? I'm not sure what is the resource that you have, and I'm not sure whether it's a reliable resources. There are a lot of research going on, but I don't think that will be the future of it. Okay, crystal clear. Okay. Okay, everyone, last question. Question number 8, and then Ashwara ma'am will continue. Right? Here we have. Give 5 points to show. It's easy, everyone. We have to give 5 points to show the significance of variation. Can you tell me what is variation? Dominating means that will be, those are the alleles that will be expressing themselves, right? They are the dominating allele, or the traits. Very good, very good. It helps in the evolution for the better survival. Yes. We don't have any official weightage for the chapters. CBSE doesn't give that. After looking at the paper and when we do the paper analysis, we have an idea, okay, from this particular chapter, this many marks can come. But again, it can definitely change. Very good, everyone. So we have 5 points and this is how you can write. If this question comes for 5 marks, so you have the straight 5 points. 3 marks question, so you can write 3 points altogether. It actually helps your organisms to adapt to the changing environment, leads to the evolution, allow the organism to develop the resistance against the harsh conditions. Beneficial variation helps to emergence of these strong species and enable survival of the organism. Yes. Yes, evolution is deleted. Control and coordinate ka PYQ is already done. Niche, when we talk in terms of biology, it's like very small, very small area, right? Niche, a very niche area. Where of course we will see a small varieties of the animals. Niche. I think how to define niche. I think in an easier way for you to understand it's that a very specific kind of thing, right? Niche area. If you go into the details of it, you just have one thing. Yes. So everyone, this is how you can write the answer in the examination. Yes, very good. Ajit, blueprint definitely comes from it, right? We have an understanding. Weightage comes in terms of the, in terms of the chapters, but in terms of the different subjects. Awesome. With an experience, we will say, okay, life process is super important, then how do is important, then genetics. Okay. So everyone, are we clear? Are we clear? Give me a quick thumbs up. Yes. Take a screenshot of this, everyone. Very good, everyone. We will have our environment also. We will have the environment session also. With this, everyone, we are done with my part. Now, Ashwara ma'am will be continuing with the rest questions. But before that, I hope that all of you have registered yourself for the All India Mock Board Examination. Right? It's for class 10th and 12th. across different grades. And it's starting from 5th to 12th of Feb. Last date of registration is 4th of Feb. You can easily give this particular test from the comfort of your home. It's an online examination. Please do register. Registration is absolutely free, right? So the registration link is there in the description box. Go click on it, register for it, and then you will be writing this in the exam. Basically, you'll be attempting an examination, right? This will be helping you to understand that, how you're writing and, what are the important points to remember. What are the points you still have, there is a small room of improvement. So it will be helping us to understand more about our preparation for the examination. Yes? Nitya, it's already done, but you can check on the channel. For the control and coordination, you can check on the channel. Yes, very good. So everyone, please make sure to register for it, right? Yes. It's a super easy, and of course the syllabus of this particular examination will be the syllabus that you all are studying. Don't have to study anything extra also. Whatever you are studying, study that and be ready for the examination. Yes. We'll meet again for sure. So we'll call Aishwarya ma'am. Yes, how many likes we have, ma'am? Oh, ma'am, please come. I'll see over here. It's for 114.
I think we can hit 150.
Yes, everyone, please make sure to hit the like button. Yes. Awesome, everyone. So I hope that you all are here. I don't think, ma'am, they need a break. We'll lower the table. In the mid-session, I was like, I have to increase the height because it was bending.
Good evening, everybody. Good afternoon, everyone. So, as you know, Ankita ma'am and I are of different heights. So, we require a different kind of setup for both of us. So, that's why it'll take us a bit of time as we switch. So, let's just give me one moment and we'll get started. Now that Ankita ma'am has clarified everything that you need, the next set of 7 questions are going to be very easy for all of you. So we will go very fast with the next 7. Let's aim that by 1 o'clock we try to finish it up, right? 20 minutes of class. We'll wind this up, the next set of questions, and we will start soon. All right, everybody. So far, how's the confidence level for heredity and evolution? I hope out of 10 you can tell me what's your confidence level. Ma'am, heredity test was there and I got full marks. Amazing, Nityashri. Amazing. It's high. Tell me out of 10, where is your confidence level at? Yes, ma'am. See, they're saying 7, 10, higher. It's at 8, 9,
nice.
9.9.
It should be 10. Come on. If it's not 10, by the end of this session, it should be 10.
Yes, and that's our aim.
Yeah, that should be our aim. Increase, ma'am, 9. Very good. Okay. So everyone, I'll meet you after some time. Please make sure you answer all the questions. And bye-bye.
Yes, full Josh. So we're going to get started. So Vishnu, can we have it ready? All right, Josh is high, then that's all that I need, right? So lots of love to all of you and we're going to get started. Now, we're going to start with question number 9, all right? So, I hope all of you are ready and I'm going to pick up the pace a little bit as I know that most questions are repetitive and you understand the pattern of questions. So, if you've understood what kind of pattern of questions you need to, what you will get and how to approach it, the rest of it becomes very simple and easy, right? So, let's have a look at question number 9, which again has 2 subparts. So, A says that what is law of dominance of traits? Explain with one example. B says that why are traits acquired during lifetime of an individual, not inherited? Explain. Now, this is a very easy question, right? I'm sure that we've solved already. We've solved very similar questions based on this. How many of you are finding this easy? Yes, very easy question. No, this is come in 2020. Ma'am, how do germ cells make a single set of genes from the normal copy? Frozen creation, please make sure that you stay tuned. I have a question towards the end. We will do that, right? Dikshita was like, ma'am, this has come in my exam. Exactly, right? So in this case, if you think about it, it's very simple. Now we know what is law of dominance. We know that when we use the word law of dominance of traits, it means that when it is in a heterozygous condition, right? Specify that. When in a heterozygous condition, dominant traits will express themselves, right? In both homozygous and heterozygous condition. But a trait that gets suppressed or which is submissive is what we call as a recessive trait. And what example would you use in this case, right? What example would you use? You only tell me. What example can we use for this? Pure breeding is when in homozygous condition, right? So when you want to get a plant which has homozygous condition, then you will do pure breeding or get pure breeds. So that is what we mean by it, okay? Ma'am, can we say that? What can we say, Aditi? I think I would have missed what you have sent me. Okay. Ma'am, are we solving sample paper during mock paper class? So no. Today evening what we're doing is a practice paper. Then we'll also be having mock paper analysis soon, right? Good afternoon. Yes. Exactly. Very good, Aidu. Very good. Exactly. So in this case, when you are using an example to explain, you can use our very own height of the plant as the character. Use tall plant when you cross a pure tall plant, right? And you can write this down. So pure tall plant when crossed with a pure dwarf plant. Yes, what will you get? You are going to get a heterozygous tall plant, and in this case we see that only the dominant character is expressing itself while in the recessive character is not. And you can draw the Punnett square to again support your answer. So when you do this, you will get 3 marks as it is, right? So this is going to be very simple and easy. Now moving on to the second one. Why are traits acquired during lifetime of an individual not inherited? Explain. 2 marks will come for this particular question also. So we've already discussed what are acquired and what are inherited. So you have to just do a compare contrast of these two. So in the chat, very quickly, you give me 1 point of difference as to why acquired traits cannot be inherited. Very good. No genetic material is involved, right? Ma'am, in heterozygous condition, one allele is dominant over the other. Can we say that? You can, but you can say that in heterozygous condition, one allele, yes, one is dominant when that expresses itself, right? So it will be incomplete when you say one is dominant over the other. When do you say one is dominant? When it expresses itself, right? So that is something that you need to do. Exactly, very good. I am able to see a lot of good answers coming my way. Well done, right? So as we know, inherited traits of course are passed on from parent to offspring and they express themselves right from birth, while acquired traits are those which we acquire during our lifetime, and in this particular case, we see that there's no change or no alteration in DNA, and again we see that hence it cannot be passed on from parent to offspring. So there you go, your answers are there. Now of course if you get this for 4 marks, normally 2 marks will come in this part where you write about Mendel's law of dominance, 2 marks when you write about the difference between acquired and inherited traits. 2 marks and a total of 4 out of 4 marks you will get easily, right? So I hope that this is clear for all of you. Now, of course, before I get started, I hope you're not facing any technical difficulties, right? No technical difficulties whatsoever. Yes, are we good to go, everyone? Are we good to go? Ma'am, side, please. Okay, yes, you can take a screenshot. Ma'am, it is tough. But don't worry. Like, it's not tough. It's simple only. Like, you tend to get a little overwhelmed about the chapter, which is why we are giving you a lot of practice. Ma'am, can we say somatogenic variation? You can, but you don't need to use very fancy terms, frozen creation, because at the end of the day, they will ask you very simple. They would want you to use simple language, right? Somatic cells are the non-reproductive cells of our body that do not participate in reproduction, right? So that is what we mean by it. So shall we move on to question number 10? Shall we move on? So we have just about 6 questions. Yes, 6 more questions and we are good to go. Very easy questions. Again, a repetitive question that is there, that is to list the differences between acquired and inherited traits, right? By giving an example of each. Now, we have already covered this part, so I am not going to spend too much time. But when you are writing it, and this comes for 3 marks, right? So, this comes for 3 marks here. You will have to be clear about it. So, 3 points of differences should be there. Now, of course, they have clearly said 2 along with examples. So, you will get 2 marks for writing difference plus 1 mark for the example. So, inherited traits pass on from parent to offspring. These are developed during the lifetime. In the case of inherited traits, they arise due to genetic variations. These are somatic variations. What are somatic variations? I know, like frozen creation was telling me, ma'am, can we do this? Of course, we know, right? In this case, you can say somatic variations or cells that you observe in the body. And of course, what are the reasons why they are caused and the examples, and there you go, right? So here 2 points of difference, you will get 2 marks, 1 mark for the example. Now in this particular question, what is it that I want you to focus on is on how to write the answer. So in this case, when you think about it, we see that you need to make a table to differentiate the two and make it clear. Alright. So moving on to question number 11, everyone, we have another one based on monohybrid cross. Okay. So a blue colored flower denoted by BB is crossed with that of a white colored flower known as BB. Right. So you have 3 subparts to which you need to identify and write the answer. Now this will easily be a 3 mark to 5 mark question that can come in your exam. Yes. So let's have a look at the first one. So first part is you have a blue colored flower. Okay. So you have blue color flower, which is BB, getting crossed with white color. Okay. So we have something which is white color. That is BB. Now what are they saying? What is the color of the flower you would expect in F1 generation? Can you tell me? Very good. It will be blue in color. And why would it be blue in color? Because they have not outright told us that blue is dominant. But because the way they have written the genotype, they have put capital letters. And because of this, we understand that all of them are going to be blue and they will be heterozygous, right? So all flowers in F1 generation are going to be blue. Now second part of the question is, what is the percentage of white flowers in F2 generation if the flowers of F1 plants are self-pollinated? So if I take this and I cross it, what is going to be my percentage? Very similar to the green stem, brown stem question that you did, right? Very similar to that. So you will do your Punnett square. You will get all of this, right? Make all of this happen. And last but not the least, you see that 1/4 is basically 25% of white flowers is what you would get in your F2 generation, right? Very simple and easy. Now, on the other hand, they're saying, state the expected ratio of genotypes of BB and BB in F2. So, homozygous blue to heterozygous blue. What is the ratio? Yes, what is the ratio for this? I am pacing it up a little because you have already solved a similar question with Ankita ma'am. It's an application. Again, go back to this. What is the question? Homozygous blue to heterozygous blue, right? What is the ratio? I am getting 1:2:1. 1:2, 3:1. Look at the question, bachas. They are asking only capital B. They are asking only 2 things, right? 1:1, 1. Yes, the correct answer is 1:2. Now, when will 1:2:1 come in the picture? When I am considering my white flower also. But here nobody is talking about my white flower. They want to know what is the expected ratio of genotypes of just homozygous blue and heterozygous blue. Then it will be 1:2, right? Now, when will you say 3:1? 3:1 is a phenotypic ratio. Okay, that is a phenotypic ratio. So in this case, you will have to write 1:2, not 1:3. Okay, 1:2. And when you are writing it, write this also. Otherwise, your answer will be incomplete. So when you're writing the answer, the first part of it was asking you what is the color in F1 generation. The color of the flower will be blue in F1 generation. Now, when the F1 flowers are self-pollinated, so if this becomes my parent, then I see that I will get white flowers in my F2 generation, but only 25% of my progeny is going to be white flowers. Then what did they ask? What is the expected ratio of genotype of capital B is to small BB is to BB, and that is equal to 1:2. So I hope we are clear with this. Are we all clear? Give me a quick thumbs up. Because see, these are silly mistakes you make in class. Okay, that is where, like now this is, see, we have identified, right? This is where you make your silly mistakes and you lose marks. Not like you don't know concept. It's not like you have not studied. You are all brilliant. Just that these silly mistakes is why we want to sit and make you practice during class itself, right? So stay focused on such things. Read the question thoroughly, all right? So now let's quickly move on to question number 12, okay? So question number 12 is a very common question. So this is All India 2016. This question has come where, with the help of an example, justify the following statement. A trait may be inherited but may not be expressed. Now this is easily a 3-mark question. Now how many of you have seen this particular question and have gotten scared saying, ma'am, how do we do this question? Or for those of you who are confident about it, how will you go about it? Now, some of you are telling me, ma'am, recessive allele. Someone is telling me, ma'am, dihybrid cross, monohybrid cross. They're saying, ma'am, it can be recessive, tall plant, short plant. Jay is like, ma'am, I'm confident I can solve this question. That's amazing, right? Very good. Ma'am, defect. Okay, we are talking about defect. What is AI? AI is all India paper, right? So, all over India. AI is just that, not the other artificial intelligence. Exactly. This is all on law of dominance, right? You don't need dihybrid cross per se to explain this particular question. Because here they're saying that a trait may be inherited but can be not expressed. So you can take the example, right? So in the case of Mendel's experiment on pea plant, when he took a tall plant, when he took a pure tall plant and a pure short plant, right? We see that his progeny in F1 were all tall plants. But that doesn't mean that this particular gene was not inherited. It was inherited, but it was not expressed. And why was it not expressed? Due to the presence of a dominant allele, right? So when a recessive allele is there in the presence of a dominant allele, it can be suppressed. So it doesn't mean that it is not getting inherited, just that it is getting suppressed due to it. So you don't need to use dihybrid cross and write the whole story about it. Use a very simple example till F1 also. If you write, it is more than enough. But if you want to add some more points and maybe write about how in F2 it reappears, you can write that point also. But the question is just simply asking you to justify this. So you can simply write it in that manner, right? So here you can write it simply in this fashion. So a trait may be inherited, but it cannot be expressed, right? And we see that this can be taken with the example of Mendel's monohybrid cross. So when a tall plant is crossed with a dwarf plant, we see that the F1 generation plants are all tall. But again, when you self the F1, we see that in F2 generation, we observe tall and dwarf plants. This goes to show that F1 plants had inherited both parental traits, just that not all the traits were expressed, right? And you can elaborate this with the Punnett square and support your answer. So if this comes under 1 mark, most often than not, this kind of a question will not come for under 1 mark. Because justify questions will come for 2 marks or they will come for 3 marks normally, right? But if at all they come for 2 and 3 marks, you don't need to write like 5-6 pages of it. Simply be very crisp about writing it till F1 generation also for 2 marks. It is well and good. So Atharv laws are law of dominance, law of segregation, law of independent assortment. NCERT does not call out these laws, but your questions will be applications of these laws, right? Yes. Okay. Oops, my mistake, guys, my mistake. It should be small t here. Yes, thank you for that. Okay. Thank you, Vaibhavi and Gaurav. I have made the change now. As you can see, the Punnett table is proper. So shall we move on to question number 13, everyone? Shall we move on? Are we feeling confident? We know the approach now, right? We know the approach now. Ma'am, law ko define karne ko aayega. Most often than not, they'll not directly ask you. They'll ask you indirectly in this manner, right? So they will ask you a little bit indirectly in terms of why did Mendel do this experiment? So very questions which Ankita ma'am took, they will ask you questions like that. They'll not really go ahead directly with it, okay? Yes, all right. Ma'am, only 1 more minute is there. I might need 5 more minutes, okay? As much as I said that I would solve it faster. But okay guys, let's move on to the next set of questions. So this is question number 13 and 3 more questions, right? So question number 13, again, easy peasy. I don't think I need to spend too much time. Why is F1 progeny always of tall plants? When a tall plant is crossed with a short plant. How is F2 progeny obtained? So we'll take it one by one. First part is, why is F1 progeny always of tall plants when tall plant is crossed with short plant? Very easy, exactly. So tallness, T is a dominant trait, right? So when you cross a pure tall plant with a pure short plant, what happens? We see that we get all tall simply because T is dominant over this. Super easy. How is F2 progeny obtained by self-pollination of F1 progeny different from F1 progeny? Give reason for this observation. So many words, okay, but basically they're saying, if I take my F1 progeny, that is this, right, and I cross it, I will get F2 progeny. Now, how is my F2 progeny different from F1 that I got? I got all tall here, right? I got all tall in F1. But what will I get in F2? Can you tell me? What will I get in F2? Exactly. Very good. See, now you're getting a hang of it, right? You're getting a hang of what you should apply at this point. Hello to everybody who has just joined. Okay. Very good. Yeah. So you see that you will see some of them would be tall plants. So you observe tall plants, but you also observe dwarf plants here, right? So in this case, we also see that dwarf plants are there. That means that by the law of segregation, we see that this has appeared back and we are getting a dwarf plant. So in the case of F1, all are tall, but in F2, we find tall as well as dwarf plant as well. Very good. Yes, very good. Now moving on to the last one. State a conclusion that can be drawn on the basis of this observation. What can we observe from this? Yes, what can we observe? Yes, Pratik, I'll tell you about the laws. Don't worry. We'll finish solving the questions first, right? Exactly, Vinita. 75% tall and 25% short. So what is it going to be? Yes, at the end of the day, everything circle backs to law of segregation, law of dominance. Now, if you get a hang of what you need to apply, right, then you are good to go. Very easy. So, what did you apply? You applied law of dominance, right? So, basically, you see that there is of course different. Observe that generation after generation, there's some changes, okay. But mainly here, what do we observe? We see that in F1 generation, we know that tallness is a dominant trait, right? And it will express itself over when in a dissimilar or heterozygous condition, the dominant trait will always express itself, right? And during gamete formation, right, we see that there has been some segregation of the alleles, which is why much later on, an allele which maybe has not expressed itself in heterozygous condition, due to segregation in, of course, the progeny, it is able to express itself. So in this case, what do you need to do? You need to make sure that you elaborate on 2 points. So what do you start with? A. The first part of it, you will start by saying that when tall plant crossed with dwarf plant, in F1 progeny, you will get all tall, right? So this means that tallness is dominant over dwarf. Now the second part of the question is asking, how is it different in F2? We have elaborated it, right? Because in F2 generation, we saw that there is tall and dwarf, 75 and 25. But F1 has all tall, right? So that is what is different. Now last part is showing us about, or is asking us, what is our observation? Now this is what I need you to focus on, right? So in this particular part, you need to write down that, see, first and foremost, dominant allele will express itself when in heterozygous condition. And along with this, we also see that at the time of gamete formation, they separate, right? The parental alleles will separate, and eventually we see that only one allele always. We see that especially in the case of a recessive condition, when they are homozygous only, they will be able to express themselves, right? So are we clear with this particular question? Are we clear, right? Ma'am, dominant will express itself on homozygous. Yes, they do. Very good, Ram. Very good. Law of segregation, bacha, I have explained it yesterday. Ankita ma'am has explained it now, but I'm going to explain it to you once again so that you understand. Think about it as, separation, right? So what do we mean by law of segregation? So law of segregation or law of purity of gametes. See, that's segregation ka another name is purity of gametes. Don't worry. Okay, suddenly everybody's seen something new and gotten scared, but don't worry. But in this case, mainly in segregation, the alleles that are there during gamete formation, it's not that the alleles will go only in one place. They can segregate during gamete formation, and based on which gamete fuses with which other male and female gamete, based on their fusion, we see that some random mixing happens, right? So that is what we mean by it. Ma'am, if it is not dominant, then what condition? It is recessive, right? Yes. We will finish it with high Josh in the next 5 minutes. I have 2 more questions with me, okay? 2 more questions. Law of purity of gametes is nothing but law of segregation, right? You will get both the PDFs for yesterday and today in the evening, right? And for those of you who have been regular to my classes, do know that I mainly teach in English. My Hindi is not very good, but wherever you completely do not understand, I will teach you that part in Hindi, okay? So question number 14, twisted version of our previous question. In one of the experiments with pea plants, Mendel observed that when a pure tall pea plant crossed with pure dwarf, in the F1, only tall plants observed. So here he is asking what happened to the traits of the dwarf plant in this case, right? What has happened to the dwarf plant in this case? At this point, you don't even need to, I don't think that you need me to tell the answer. You only know the answer, right? You know the answer. We have done monohybrid cross, law of dominance so many times, right? Very good. It has gotten inherited, right? But not expressed. Now, this is incomplete. So you need to justify. You need to justify why it has not gotten expressed. Yes, very good. Should we learn about Mendel? Mendel's laws you should definitely learn. Okay, very good. Ma'am, in NCRT there is no mention of allele. So if we write it in the answer, you'll not lose marks. But see, instead of alleles, you can say traits also. Because at the end of the day, alleles code for your traits, right? So if you say dominant trait, recessive trait also, it's fine. Exactly. So in this case, we know that dwarf plants is a recessive trait. Yes, and we know that in heterozygous condition, it will not express itself because the dominant trait will express. So you should write, okay, you should write it fully. Half and half you write, your marks will be gone. Second part. When F1 generations were self-fertilized, he observed that in second generation, both tall and dwarf were present. What happened? Why it happened? Explain briefly, right? No, 2 more questions, Avika, and we are done with the session. We just have 15 questions, right? Now again, we know the answer to this, right? The answer comes from the law of segregation, right? So during formation of gametes, we see that there has been segregation of these alleles, and as a result in the F2 generation we see that a character which was earlier not present or a trait which we did not observe in F1 has come or has reappeared in F2, right? And we know that recessive traits will only express themselves in homozygous condition. Can we explain this using Punnett square? Of course you can explain using Punnett square, whatever is comfortable for all of you, right? So with this, of course, you see these kind of questions. It's a very easy question. So most often than not, this can come for 2 marks or it can come for 4 marks. But if it does come, you will have to elaborate on it, right? Very good, very good. So you can write the answer in this manner and you can finish it. Like you can conclude your answers as well. Now moving on to last question. We are clear with the previous question, right? I'm not spending so much time because we've already discussed it in detail. But I just want you to get a practice of the question, which is why I hope you're comfortable. Yes? Are we all good to go? Give me a quick thumbs up. Last question for today. Yes. Need the enthusiasm super duper high, crystal clear, then that's all that I need, right? So before I go into question number 15, everybody, if you enjoyed today's class, you found this helpful, you felt like this particular class really helped me, please go ahead and hit the like button on this video and do not forget to subscribe if you are new, because you know that in BYJU'S 9 and 10, we go out of our way to make sure that you ace your exams, right? Because it's almost as good as us giving the board exams. All right. Ma'am, compulsory we have to write Punnett square. See, like I said, if you have 2 marks and they're just asking you to state something, you don't need to drop Punnett square. 3 marks, 4 marks, 5 marks, advisable to drop Punnett square because it will support your answer better, right? So that is something that you need to do. All right. Okay. So now moving on to question number 15. Now this is a question some of you asked me yesterday also, for those of you who have attended the crash course session. In case if you've not attended that, you can go check it out. It's there on the channel. But a lot of you asked me this question on explain how equal genetic contribution of male and female parents is ensured in progeny, right? Now, I'm sure a lot of you here, right? How many of you have got this in your pre-boards? How many of you have got this particular question in your pre-boards? Or explain the manner in which the sex is determined? Yes? Quickly give me a thumbs up in the chat telling me, ma'am, yes, this question has come. Or something of this sort has come in my exam. Yes. Oh, Harshita's like, ma'am, no, this didn't come for me. It's okay. In a way, it is good if it hasn't come. But if it has come, there's nothing to worry about also. See, they have twisted the question. Exactly. So, this is a statement which has come off NCRT. Okay. This is a statement as it is from NCRT textbook where they say that during gamete formation or during the formation of zygote, we see that there needs to be equal genetic contribution, right? Equal genetic contribution of male and female parents. Now, why is this necessary? Now, we know that in the human body, all cells have a fixed number of chromosomes, or we have 46 chromosomes, right? And we know that human beings are sexually reproducing, which means that we produce gametes that will come together and fuse and result in the formation of a zygote. Now, the number of gametes the number of chromosomes in your gametes matter, right? Because we know that formation of the young one starts with fusion of gametes, which is why we see that half the number of chromosomes exist in both male and female gametes. So 23 and 23 exist. Now, why should there be 23 and 23? Because at the end of the day, these 46 chromosomes or these 23 pairs are formed in a way that one set comes from mother and one set comes from father. Which is why at this point we see that during fertilization, we see that there needs to be equal contribution. So 23 plus 23 will give you 46, resulting in the formation of a zygote. And in this case, you can write it in this manner also, 22 plus X or 22 plus Y. Here again you have 22 plus X. And again, based on the kind of sex chromosome that is there, you will be able to get it, right? Exactly. Ma'am, can you explain genotype of green-stemmed plant, tomato plant? Musical shorts, if you rewind back, the second question Ankita ma'am has done is a sort of a similar question to what you are asking. You can check out that particular video, right? I mean, you can check that part of this video. It will definitely help you out. So all you need to do is to write that, right? So you need to write two parts of it. You need to mention the sex chromosomes part and mention half and why haploid is necessary, right? So this is the answer. And as you can see, you have to write it down in elaborate points of this sort so that you will be able to understand, right? And we see that in this manner, the sex of the child can be determined. And we see that, you know, if the question is oriented towards sex determination, mention the point on how, right, it helps, right? Especially, so this is for actually the first part of the question. Explain the manner in which sex is determined. That is the answer. But if you are getting the second part of the question, that is how is equal genetic contribution taking place, that is what the explanation that I gave you, right? Ma'am, if they ask diploid, can we explain? Yes, you can. Diploid is basically what? 46, right? When you have two sets of your chromosomes, that's your diploid condition. Do we need to explain the laws? Like I said, they may not directly ask you to say state law of dominance, but indirectly they will ask you, right? Okay. So what is diploid haploid cell? I'm going to take a minute to explain this. So see, we have 46 chromosomes or we have 23 pairs of chromosomes, right? So when we have 23 pairs, that means that they are all in pairs or what we call as 2n. So this condition where they exist as pairs is what we call as diploid condition. But at the same time, when they don't exist as pair and you have only one half of it, right, it becomes a n condition. Now you have only half. This means that it is haploid. Haploid again means half the number of chromosomes, right? So that is what we mean by it. So are we clear? Are we clear with this? Ma'am, can you explain blood group based questions? I did a few yesterday in class, but if you feel like we need to do more of blood group based questions, let me know in the comments of this video. We'll definitely convey it to our team. And as you see, we will later on in the weeks to progress, we will be doing more case based questions, assertion reason based questions. So don't worry about it. We have got you covered quite literally. So all we need you to do is to stay subscribed to our channel, right? Very, very important. Ma'am, from one cell, 4 gametes. Yes, in the reproductive cells, there will be one mother reproductive cell from which you will get 4 gametes, right? And again, one last time to recall Mendel's law, right? Mendel's law of dominance suggests that in a heterozygous condition, if there is one trait that expresses itself over the other, this trait is called as the dominant trait, and the one that does not express itself is called as the submissive or the recessive trait. Your law of segregation suggests that during formation of gametes, there will be separation or there will be segregation of alleles during it, right? And your law of independent assortment says that when you consider more than two alleles, right? We see that it's not like they will segregate together. They will independently assort, or they will independently separate, and it's not based on any certain factor. So this is what we mean by it. And of course, for those of you who have just joined, I've already finished the session, so you can go back, start from the beginning where Ankita ma'am has taken the class, right? So everybody, here's a reminder for all of you on BYJU'S National Scholastic Test and Personality Test. It's happening for free, and the last set of dates are on 4th and 5th of February. So link is again there in the description below, and the advantages again of you joining this test is for you to get some career inclinations, some capability analysis, overall analysis, so on and so forth. Link is there in the description below, and you stand a chance to win scholarship for BYJU'S Tuition Center. So check it out. And of course, Ankita ma'am, with this we come to the end of today's class.
Yes, everyone. So I hope that this session have helped you in understanding the different types of questions that can come from this particular topic. So please make sure, if you're joining just now, please make sure to go through the session again, and please make sure you watch this session after watching the main session which ma'am did yesterday. Okay?
Yes.
So it was a detailed class. So watch that session, and then this. It will be all sorted.
And everybody in the comment section below, let us know what is your confidence level for the chapter, right? Confidence level out of 10 is what we want to see. Let us know in the comments of this video. Ankita ma'am and I will definitely be checking out.
Yes.
And see, you have both of us, right? We are both here to hold one hand of yours to make sure that you ace your exams.
Yeah. So don't worry. We still have a lot of time, and we will be, till your examination, till the day of examination, we are here. In the examinations, our blessing will be there. We cannot be there with you.
Alright, so with this, we'll be signing off, everybody. I hope all of you have liked the video, shared this video, and ma'am, subscribe to our channel, right?
Yes, please make sure you do all of these three, because it will be great for all of us. So on that note, everyone, I think we'll say bye-bye. Everyone, do take care of yourselves and keep on learning with BYJU'S.
Bye, everybody.